Thank you so much for that kind introduction, and Tal, thank you so much for the invitation to speak with all of you today. I'm so excited to be here. We are, at the MMRF, huge believers in the precision medicine model, and it's really because we see three incredible trends in healthcare taking off, and precision medicine tends to bring all of them together. So we look at the science of genomics and immune therapy, and we combine that with the big data and digital revolution we're all seeing. But we would be remiss in not mentioning the patients and the consumerism we're seeing, where patients are really starting to understand and share their data as much as they can. The other reason that we're so passionate about this space is because we find that many of the precision models that we're starting to see, and there are many sprouting up, are also allowing us to integrate all the different groups in the healthcare continuum, from diagnostics to academics to pharma companies and FDA. I was very fortunate over the summer to work with Kathy Hudson as a member of the Obama Working Group on the precision medicine model. And it does seem that a million-patient cohort will certainly teach us a lot of new things on how do we attract patients and even just consumers to the fold? How do we encourage them and consent them to give as much broad data as we possibly can? How do we inspire them to stay involved in a program like that for as long as possible so we can garner as much information as possible? I think California is doing an amazing job, which is the reason we're so excited to be out here as well. And then, of course, other programs like what we're seeing in Ohio and with Eric Dishman at Intel. So again, so many different models that we tend to study at the MMRF and try to follow because we know we can learn from them. But today, I was asked to come out and talk with you about the model that we've developed at the Multiple Myeloma Research Foundation. And I talk about how we developed it, but please know we developed it with the entire myeloma community behind us. So multiple myeloma, for those of you that aren't really familiar with it, it is a blood cancer. It is highly fatal. It is highly heterogeneous, and it's uncommon, and it skews older, male, and African American. I was diagnosed with multiple myeloma when I was 37 years old, and at the time there were no drugs in the pipeline, and it really was pretty much a death sentence. But I thank all the work at the MMRF, and I thank the entire community that started working with us, because the results in this fatal cancer are now astounding. Just think about this. Since we started our foundation, we have seen 10 drugs approved in multiple myeloma. 10. We've improved the 5-year survival by 40%, and patients no longer live that very sad 3 years that I got when I was diagnosed, but now it's more like 8 or 9 years. So the point is, we feel that if we can understand precision medicine and models like this in a very uncommon, heterogeneous, fatal cancer like myeloma, the truth is we probably can figure out precision medicine and ways to have progress like this in all cancers. So our model looks like this, and really it's been 15 years of work laid out in 3 simple blocks. And I'll walk you through them, but just to understand, first for us is our data bank. So everything we do at the MMRF is all about big data. We started with our own genomic initiative in about 2004 that we were doing with the Broad Institute, and it was based off of one-off tissue. But having done that, we also realized the importance of collecting genotypic, phenotypic data and doing it longitudinally. So we launched a $40 million initiative called Compass, which I'll talk with you about a little bit more. And once we started aggregating all of this data into our data bank, the most important piece was saying, well, okay, what are we going to do to put that data into the public domain, and then also make sure that other people were analyzing it? So within our learning network, that's where we tend to work with companies like Genospace that houses our data in a gateway and pushes it out for all the scientists to see. And then we also work with companies like GNS in Boston, where they take the raw data and analyze it to help us understand the pathways in myeloma. We then take all of that information and we drive it to the clinic. At the MMRF, we have our own clinical network of 22 centers that have all signed membership agreements with us. And those 22 centers do phase 1 and phase 2 trials with us. They're held to metrics: how fast are they opening, how fast are they enrolling, did the investigators bring new ideas, and together, at any one time, we usually have about 15 to 20 phase 1 and 2 trials going on that we're helping to spearhead. So that helps you to see the model in 3 simple blocks. But the important piece and the cornerstone of this model, which is so important to everybody in precision medicine, is what we did with the COMPASS study. So the COMPASS study was launched 4 years ago, and at the time that we decided to take this on, everybody said, there's no way you're going to get this done, because our goal was to sequence 1,000 patients, but to sequence them at the moment of diagnosis. We wanted to make sure we were capturing their genomic sequences before they started treatment. Then what we do is we collect all the clinical data on these patients, and when they relapse, we sequence them again, so that all of the longitudinal data is captured into our database. And what you can see from this slide is that we're studying this over 10 years. There are 90 centers that have been involved. We also, because it's a $40 to $50 million initiative that our foundation was taking on, we had to build a pre-competitive consortium. In that pre-competitive consortium, half of the funding for COMPASS came in from philanthropy, and the other half came in from our pharmaceutical partners that got an early look at the data. But you can imagine the data, based on all of the testing that we were doing amongst these patients and that we will continue to do, is very robust. As a matter of fact, one of the largest data sets in genomics today, which is pretty amazing when you think of how this cancer skews and how uncommon it really is. So as we move forward in this model, and just to give you a little more clarity of how it works, I talked with you about a small genomic initiative we started with just one-off data, and then we built on that initiative with the COMPASS study with over 1,000 patients longitudinally. Now we're sequencing 500 patients relapsed through an assay at University of Michigan. Mount Sinai has a program going on. Foundation Medicine has a data set of myeloma patients that we're in discussion with them about as well, and then we're evaluating a registry. These are all the areas that at any one time we're trying to negotiate to make sure we're building the largest data set that we possibly can and integrating the data where we can. Now, we know there are lots of other data sets out there in myeloma that our academic centers are holding, that biotech and pharma may have, and registries. You know, our goal at some point in time will to make sure that everybody comes together, even if it ends up being some type of a federated model. But our hope is this is going to be the best way for us to find cures. I think one of the greatest areas for us, though, is now that companies have found out we have this robust, pristine data set, the joy is having other partners come in and analyze the data set with us, which is phenomenal. So Genospace, as I said, actually captures our data and pushes it out to the public domain. But GNS Healthcare has done a phenomenal job, and I'll show you some slides later on the work they're doing with us, where they literally take the raw data. They do what's called the data jamborees. They work with all of our academic centers. We share everything they're finding on their algorithms back with academia and industry, and we put that data back into the data set for everybody to use. So again, having data analytic companies like that working in our model is absolutely the wave of the future for us. And so we continue in discussions with all of those groups. And again, everything we do has to be driven to the clinic, otherwise we're no longer serving the patients that have a fatal disease. So just to give you an example of how the MMRF became an end-to-end solution, even when we were just starting with our first genomic initiative, back then, working with Todd Golub and the Broad Institute, we sequenced 38 genomes, and we found that BRAF was a target in multiple myeloma, obviously a known target in melanoma. So that was published in Nature by our scientific team and others. And then obviously knowing venurafenib was out there, we tested that drug in myeloma patients. Responses were seen. And then we went back again and looked at our genomic initiative, and we continue to study this through COMPASS. And one of our scientists, a gentleman, Daniel Leclerc, who's in the audience, helps us to continue looking at these pathways, and we found out it's really BRAF and MEK. And as a result of that, we're driving that to the clinic with a combination trial this quarter. So it gives you a sense of how we make sure the data we're aggregating gets analyzed, gets validated, and gets driven to the clinic. Now, the important thing in myeloma, as I said to you, is it's highly heterogeneous. So BRAF is only 5 to 8% of our patient population. But based off of all the data that we've already accumulated, 50% of our patients have some form of a mutation for which there's a drug or something that's probably in the pipeline or clinic. So everything we work on right now is making sure we speed that precision medicine model that we've been showing you so patients don't have to wait to get the right drug for them. The other space that we continue to work on is how do we create much more innovative and efficient trial designs. It's really hard, as we all know, to do these small, very precision-based trials, but if the MMRF is willing to pay, and we often are paying for this, for the sequencing of our patients and then moving them in to master protocols like this one, it makes it a lot easier to bring our pharma partners in. And it also makes it a lot easier for us to play in a space that we love, which is sitting down with FDA and working through new regulatory strategies that might be more efficient for all of us. So innovative trial design is a place that we actually put a fair amount of money in and make sure that we're really focused on it. The other piece is somebody has to find all of these patients. So that's another area that we play, is collecting all of the patients, knowing what they have, educating them on their genomes so that we can find them and enroll them in the right types of very precision-based trials. So I've given you a little bit of an understanding of how we work as a virtual biotech company and take precision medicine. We're driving new drugs to the clinic and trying to have as much progress as we possibly can in that field. But we are still in touch with patients every single day. And we know the questions that our patients and our community oncologists all have. And it's our job to make sure we answer them. So we always say to people, like, big data is wonderful and it's great, but if you aren't answering questions, or if you don't know what questions you want to ask next, it's really not helping you that much. So one of the spaces that was concerning us in myeloma was we had many patients that were on a doublet of a proteasome inhibitor and DEX, or they were on an IMiD and DEX. But the question was, were we all better off doing a proteasome inhibitor, an IMiD, and DEX, and being on a triplet? The reason it's important is obviously expense. It's also side effects. But patients do want to make sure they're getting the best treatment up front. And this was data that was provided to us by working with GNS once again. And you can see, based on these curves, that yes, now we can actually answer to our patients and community oncologists, you should start your patients on a triplet. But in addition to that, another major expense and another hardship for patients that have multiple myeloma is, should I do a stem cell transplant or should I not? And so when you have 10 drugs that have now been approved in the marketplace, people are trying to decide what is the role of transplant. Well, indeed, there still is an incredible role for stem cell transplant in the field of multiple myeloma. And as we continue to move forward, what we'll do is more genomic work to make sure we're understanding exactly what the patients look like that are benefiting the most from stem cell transplant. To give you another example of how we work with GNS is, there were a lot of small studies on cyclin D1 inhibitors in myeloma. We weren't seeing everything that we wanted to see. But now, based off of their data, it does look like that's an area that we should pursue. So we want to go back now and study that space and say, is there something else we should be doing, looking at a patient population, looking at other drugs, so that we don't walk away from categories that actually might be important to us. So I've mentioned these are all the things that we're answering today. But as a team, our executive team sits down with all of our scientists and partners, and we say, what are the questions that you really have in the future? Because if we're going to invest in sequencing and paying to build our data bank and working with data analytics companies, what do we need to know? And this is how we end up working on our business plans. It's what are the questions out there? So 10 drugs approved, some amazing antibodies. Now the question is, should we be starting patients on 4 drugs instead of 3? What is the role of the antibodies in immune therapy in myeloma? So we need to go back to our data banks and also look forward to what we're collecting to understand how to answer that question. A huge area in multiple myeloma is patients are now living longer and more patients are getting into complete remission. If that's the case, should everybody really be on maintenance therapy? If you're left on maintenance therapy for 4 or 5 years, the expense of that, along with the side effects or even other potential risks, has to be managed. So what is the role of MRD in understanding who should go on maintenance and who should not? We have to imagine that's a really important question for patients, for the oncologists, for the payers all alike. So we have to garner that information. And then, of course, every patient asks the same question: if I relapse, what do I do? The disease is still fatal today. So, of course, we're empathetic to that as well. So I want to talk just briefly, because the patient is a critical component of this model, and understanding the patient piece is something that we focus on every day. So to give you an understanding of a program that we just recently did this year, quantitatively, we interviewed patients that were very familiar and educated by the MMRF, our organization. And then we also looked at another group that were not educated by the MMRF, had no awareness that we were out there. And then we put together a 12-step journey that we thought was very important for patients to go through, and we tried to find out how much more educated and willing the MMRF educated patients were to take the right journey. The next set of slides are pretty interesting. For those that are MMRF educated, looking at banking their tissue, over 3 times more likely to do it. Genomic testing, twice as likely to do it. Willing to share their data, much higher. And while we still have some work to do in terms of getting them into the trials, I think as we educate them on what your genome looks like, what precision-based trials are out there for you, this space is going to really explode for us as well. So today I've talked with you about the model. I've talked with you about the role of patients, but sometimes a picture says 1,000 words. This photo is a picture of myself, my daughter, Nicole, who's 21 years old, my 85-year-old mother, and my identical twin sister, Karen. The things I want you to get out of this is a piece that we actually did for the Wall Street Journal just recently. If you haven't seen it, I really encourage all of you to go back and read it. We decided that as a family, we wanted to get our germlines tested. And the reason for that was looking at this group. I have multiple myeloma, got it at 37 years old. Thank God my daughter is healthy. My mother's been battling melanoma for years. And this past year, my identical twin sister was diagnosed with stage 3 breast cancer. So the reason we wanted to get germline testing done was our grandfather had multiple myeloma. Our grandmother had breast cancer. So it's always curious why 2 identical twins get different cancers, and what should you do about that? But we also thought it was really important for us to be public about a journey like this, because it's what really helps the research process and helps patients and family members to understand scientists need tissue, scientists need data, and we all have to be part of that solution as well. The other piece of this picture that's really important for you to know is when I was diagnosed with multiple myeloma, our daughter Nicole was 1 year old. I wondered if I would ever see her go to kindergarten. And this year I will watch her graduate from college. And I wonder, thank you. And while I always prayed myeloma running in our family, I started this foundation to be there for her and her brother that I had after I got diagnosed for as long as I possibly could. But I never wanted the next generation to have to face a disease like this, and I worried about it. But I have to say now I'm very focused on MGUS in myeloma, or smoldering patients in myeloma. And the focus for us really as we move forward is not just treating people in precision medicine, but preventing people in precision medicine. And I think that's becoming really important to us at the MMRF. And the final piece is when your father dies of kidney cancer, your mom has melanoma, and your sister has stage 3 breast cancer, and you know you have a model that works, the most important thing we can do at the MMRF is share it. Share it with anybody that wants to hear about it. And so I'm so humbled to be here today with all of you that have done such amazing, amazing work in precision medicine. I was so impressed with everything I heard yesterday. So on behalf of our family and everybody at the MMRF, I want to thank you for everything you're doing. And I want you to know that we're here to share our information in whatever way we can to help all of you and all of the patients we serve. Thank you so much.